All right, welcome everybody to this ViQi webinar. We have over 100 registrations today, which I'm really excited about. My name is Lyndall Fraser-Cairns, and you're in the right place if you are here for practical applications for AI in high content analysis. I'm joined today by Ilya Goldberg. I will stop sharing my screen for a second here so you can see his face. Hi, Ilya.
Hi, everyone. Thank you for joining.
All right, a few logistics. If you are unfamiliar with Zoom and you're having some audio problems or some video problems, please try the old IT trick of shutting it down and opening it up again. There is a Q&A functionality in here and a chat functionality. So if you have questions for Ilya at any time, please put them in the Q&A section. And if you have problems or you want to share your experiences, feel free to use the chat. We will be sending a recording of the webinar by email afterwards, so watch out for that. And enjoy. So we are ViQi. If you're not familiar with who we are, we provide large-scale image analysis and visualization expertise and cloud-based software for scientific researchers and developers. We visualize, analyze, annotate, and store image and data formats. We automate and scale through AI, and we have the ability to tap into a team of experts to solve complex image and data challenges. This webinar is part of a three-part webinar series. If you haven't seen the first one, Introduction to AI and Classification, check out our YouTube channel. We are here in the middle talking about dose response today, and we need your help with what our future webinars should be. So when this webinar closes out, please take a minute to do our survey. It will help us figure out what the next topic should be, as well as giving us some important feedback on how you enjoyed today. After this webinar, we'll be offering no-cost consultation sessions with Ilya to help you meet your HCA goals. Our ultimate goal is to create a product that meets the needs of experimental biologists doing high-content analysis. We want to continue the dialogue, so watch out for our email about this. All right, let's start off with a poll. I want to know, what role does AI play in your research right now? Panelists can vote on this, so feel free to do that, Ilya and Kathy. You should see the poll pop up very soon. Awesome. So a pretty good spread across each of these categories with a lot of people that don't currently use AI. So I'm really excited to hear that you're excited about this, and Ilya will take you from don't currently use to extremely excited. All right. So I want to introduce you to Dr. Ilya Goldberg. He is our Chief Science Officer. Dr. Goldberg has a long career at the intersection of biology, imaging, and AI. Ilya co-founded a company that developed the first medical device cleared for using an AI to predict malignancy in lung nodules using CT exams. The research group he led at NIH developed machine learning software for image processing in biology and medicine. At MIT, he co-founded the OME project, which is still used for imaging infrastructure in large image repositories. And he has over 60 peer-reviewed scientific articles from his time at Johns Hopkins, Harvard, MIT, and NIH in molecular and cell biology, pattern recognition, image informatics, and the basic biology of aging. He is ViQi's Chief Science Officer, and I am excited to learn from him today.
Thank you, Lyndall, for the nice introduction. And thank you all for your hard work putting this together and for hosting today. I'd also like to thank the ViQi team, especially Dimitri and Chris, for all their help. Today, Lyndall and I will discuss the benefits and potential of using AI in dose response assays. So a big thanks to Molecular Devices for the data we'll be using in this webinar. Our agenda today is to learn three things: train and evaluate AIs for scoring continuous morphological process, score a multi-compound screen based on a dose-response standard curve, and then score any type of phenotypic response without knowing what to look for a priori.
Awesome. So this is designed to be a fairly practical webinar. So we hope that you'll chat questions in the Q&A box for Ilya as we are talking. We're also trying a more innovative interview format for this webinar. So I'll lead off with a question, if that's okay, Ilya.
Absolutely.
So Ilya, in the previous webinar, we focused on how AI can help with classification problems such as live versus dead cell identification, a qualitative assay. But what about quantitative assays like dose response? How do we use AI to assign a continuous variable rather than just categorizing things as being one type or another?
There we go. So even in a qualitative assay, for example, a case control study where you have affected and unaffected cases, or let's say benign versus malignant, an AI can report the probability of an unknown sample being malignant, for example. Typically, AIs report probabilities for all of the classes that they were trained with. So here we're seeing in one case, a benign case, the AI reporting 85% being benign and 15% being malignant. And with a malignant case, we see 10% benign and 90% malignant. So these probabilities can be interpreted directly as a risk of malignancy. So this is a way where just even in a binary class problem where the problem is set up as a categorization, you can use the primary outputs of an AI to interpret a continuous variable directly. And most types of AIs, modern AIs, do report these probabilities in their outputs, even though mostly we think of AIs as classifiers, and certainly that's how we introduced them in the last webinar.
Okay, so that's how an AI output can be used in a case control study and interpreted as risk. But what about cell-based quantitative assays like dose response, where we're trying to measure an effect across multiple concentrations? How does that work?
So I'm going to use this webinar to describe 3 different ways of scoring dose response assays using AIs. We're starting from the simple to the more and more complicated. So first, let's introduce how a typical dose response experiment is set up. In an HCA format, you would plate cells on an imaging plate and then apply multiple concentrations and multiple replicates of each drug to the plate wells. Typically, you'd use 5 to 6 concentrations for each drug and then 3 to 5 replicates of each concentration and drug, and then do that for as many compounds as you like. Then you process the plate fluorescence imaging, typically, and image it on a plate scanner. So here's an example of what an image like that would look like. Here we're looking at the effect of compounds on neurons. And so to keep things simple, we're only going to look at 2 channels today. DAPI is typically the nuclear stain in blue, and FITC, which in this case is a viability stain, which is in reddish pinkish. So in this assay setup, living cells have both nuclei and FITC, while dead cells have only nuclei. And you can see examples of both in the image here. So like we did in the previous seminar, we can train an AI to recognize live cells, dead cells, and debris just by clicking on them and training an AI classifier to score the rest of the images. So just using this, we can set up a pretty simple quantitative assay where the readout is just the percentage of live cells. So here's a view of an assay like this. Initially, so in this specifically, if you were just looking at the wells arranged like they were on the imaging plate and a heat map overlay representing concentrations of the various compounds. We can use this to click on wells to examine the images that were taken there, how many images there were, any kind of quality control issues, etc. Since it's a virtual plate, we can rearrange it to look at compounds versus concentrations instead of rows versus columns. And we can use a heat map to show our viability assay results. We can see the compounds that kill the cells fairly effectively. They're the ones that turn red at the bottom. For example, like mercury on the far left is a positive control, and you can see that it starts killing cells off fairly early in this concentration profile. The one right next to it is the negative control, and that's just DMSO. You can see that there's all of the different concentrations have the same effect, which is the same viability that we started with.
Okay, so mercury's bad for my brain. Got it.
Yes, we already knew that.
So as long as you can recognize them manually and train the AI, you can expand on the viability assay to other cellular effects?
Yes, so that's definitely a way to do this. We can expand this, for example, to look for binucleate cells. So this is an important target phenotype for potential chemotherapy. And the reason is that dividing cells, if they have errors in mitosis, then typically they end up with having multiple nuclei. And so this is a valuable target phenotype for screening potential chemotherapy agents. So if we just proceed as we did before with the live dead assay and then click on the binucleate cells and then train an AI to recognize them, then we can, again, just count the percentage of binucleate cells and use that as the assay readout. This is an example, by the way, of an assay where the target phenotype is important, like I said, but the development of the assay using traditional image processing techniques was very difficult. It's pretty easy to see just by eye the difference between the normal and binucleate cells, but it turns out that it's actually fairly difficult to describe this specifically enough so that an algorithm will be able to recognize these cells accurately. As it turns out, this is a trivial problem for AIs. And in fact, in my lab at the NIH, this phenotype held a record for being the most accurate AI you can train with the fewest number of samples.
Okay, so if we already have a good way of recognizing binucleate cells, what's the advantage of having an AI?
Well, so this is only one assay, and it was very difficult to develop. It wasn't just clicking like it is with AI. Usually that's normal for developing assays using traditional image processing techniques. They take a lot of effort, and even when you take the effort to develop them, the user of these things is still left with fiddling a lot of parameters to make the algorithms work for the specific imaging setup, the cell type, etc. So a huge advantage that AI has over conventional techniques is that you don't need to tell the AI what to look for. Instead, you train the AI by giving it examples of things that you consider to be different, and it's the AI's job to figure out how to discriminate between your examples.
Right. So let's say we don't really have a target phenotype like binucleate cells, but instead we want to compare several drugs to a drug that's already well categorized.
Sorry, you're breaking up there.
My apologies. So if you don't have a target phenotype, like binucleate cells, how about if you wanted to instead compare several drugs to a drug that's already been well categorized?
Right. So if you have let's say, model compound, right, you would just train the AI on a standard curve of this model compound, and then you would use this trained AI to score the other compounds.
Okay, how do you do that?
So just like any standard curve, you set up a standard set of drug concentrations and use each one as a separate class to train the AI. The difference here is that there's no clicking on cells. You just label all the cells in a well based on the concentration of the drug in that well.
Got it. But then won't the AI just tell you which concentration in your unknown sample is the most like? How do you get a continuous variable to score cells that have intermediate effects?
Right. So as I mentioned before, most modern AIs will report a probability distribution for the class as you defined during training. So a sample will have a probability of being in class 1, a probability in class 2, etc. And so here you can see a single sample being assigned marginal probabilities by the AI in the second column. And we're calling them marginal probabilities, by the way, because they all add up to 1. So the way that you combine these into a one variable, one continuous value, or in other words, a score for the sample, is you simply multiply the marginal probability from the AI by the numerical value of, for example, the drug concentration or whatever numerical value you've used in your classes. And then after you multiply them all out, you add them together and you get essentially a weighted average. It's weighted by the AI, and this weighted average is a score for your sample. In this case, because we're trained the AI on drug dose, this score would be an equivalent drug dose for that particular sample. There are also AIs that are actually regressors rather than classifiers. So in other words, you train them using a continuous variable as opposed to discrete variables like we've done here. And you get a continuous variable back out. But typically we deal with discrete variables in our experiments. And so we use classifiers and their marginal probabilities to determine scores for our samples. So let's do a walkthrough of what training an AI like this looks like. So here we're looking at the user interface for ViQi for training these kinds of AIs. And what we've done is we've selected a group of images that represent a dose response curve for a specific drug. And I want you to notice is what I've zoomed out here is the parameters that we're using for this run. So we need to specify the annotation level. So here we selected image-based annotation because, like I just mentioned, the cells are labeled by whatever concentration is in the well or in the image, rather than the label is associated with individual cells. So instead of choosing cell level annotation, we chose image level. And the label that we're after is concentration. So it's the concentrations associated with the images that we want to use to define our classes in these images. We also want to do something else because we trained this AI previously, not this AI, a different AI previously to discriminate between live and dead cells and debris. And here we want to eliminate all of the objects in the image that are classified as debris by the previous classifier. So we select the label that we want to discard, the debris, and then basically all that's left is the checkboxes that say that even though we're doing training, we actually want it to label the images with the results. So the important thing to note here is that we're not telling the AI about what differences to look for. And also very importantly, we're not specifying anything about the AI training either. This is a fully automated process that looks at many models, scans various parameters, and uses best practices to evaluate the AI and produce the scores. We've seen this with the cell classifier in a previous webinar, and this is essentially the same thing. The difference here is that it notices that our class labels are numeric because they originated from the concentration image annotation, and it will therefore compute continuous variables for each cell it encounters as well as class assignments.
That's pretty cool. How do we evaluate these quantitative AIs? Like, what does it look like when it's working?
So an important tool to evaluate AIs is again the confusion matrix, just like we've seen for classification. And here we're still looking at classification results. So in other words, we're looking at the results of the AI's calls of putting the cells into each of these bins. So we're not dealing a continuous score yet. We'll be looking at that in a sec. So what I do want to point out is what's new here. So the confusion matrix, as before and always, it's arranged with the known classes in rows and the predicted classes in columns. When we have numeric classes, we also want to arrange them in order. So the first row tells us how all the samples belonging to the first class, 0.03 concentration, were classified by the AI into all of the other classes. So most of them ended up in 0.03, but quite a few in 0.1 and then 0.3, and then it kind of dribbles out. And then you can follow that row by row all the way down. And what you want to see is that there's a strong diagonal here. So I've basically color coded the number of samples with red, so you can easily see where most of the samples are. And so an AI that's working perfectly, it would only have samples on the diagonal because it would never be confused about anything. This is why it's called a confusion matrix. However, if it's getting things wrong, right, then what you want to see is that it's not putting the samples into bins that are far away from the correct sample, right? So because we know here that concentrations that are similar to each other should have similar phenotypes, and so we don't want to see the AI confusing a concentration that's very different from the one that it's supposed to be assigning. So the key thing to look for here is a strong diagonal and for samples to be piled up around the diagonal, not scattered all over the matrix, right? The other thing that's really important to note here is that we never told the AI about the order of these classes, right? So it's just assuming that each class is different from each other as every other class, and there's no inherent order or this neighboring classes idea. And so the fact that you see this in the confusion matrix is a very important validation that the AI is actually working and picking up on real phenotypic differences between these drug concentrations. So this is just discrete. As I said, let's look at the continuous scores using box plots. So box plots are a familiar way to look at the results of these dose response experiments. And these graphs are in the output of the trainer whenever there is numerical classes. To summarize, the scores that it gives to each of the cells. So each dot that you see that's an outlier here corresponds to one cell. The x-axis is telling you the known concentration of the drug that the cell was exposed to, and then the y-axis tells you what the score that cell got from the AI. And so you can see for the positive control mercury and for rotenone, which is another positive control, you see a increase in scores as you increase the drug concentrations. And you can see a difference in effects. For instance, mercury seems to have some fairly significant intermediate effects, whereas rotenone seems to not do anything until it gets to a high enough concentration, and then it snaps up pretty quickly. So this you would say is a more classical kind of biphasic distribution of dose response, what we're seeing in rotenone. On the other hand, mercury is a kind of a good candidate to use as a model drug because it has kind of this linear scale. So it's a good candidate to use as a model drug, this AI, to score other drugs so that they would fall on a more continuous scale versus the more distributed scale for rotenone.
Okay, what happens when you don't have a well-developed model compound?
Right, so you can use a similar strategy we've used here, but instead of training an AI with a control compound and then using it to score the rest of the compounds, we can simply train a separate AI for each compound. Each of the trained AIs would only be used to score the compound it was trained on. This way, you're not comparing the compound to something else. You're only comparing concentrations of the same compound.
But you can't have the AI score the same data that it was trained on, can you?
Not exactly, no. Right, so we have different cells in these wells that were used for training that we can then use for testing. Of course, and as diagrammed here, typically you would reserve 20% of the samples or so to use for testing or scoring the trained AI and 80% to train the AI. Of course, with this kind of setup, it's a bit unfair because the cells are in the same well and the conditions are potentially different in a different well, even with the same drug and the same concentration. So in this type of scoring, we're not requiring that the AI be reproducible across wells. This can be okay though, because we're not necessarily interested in a reproducible AI at this point. Potentially, we're only interested in results from different AIs being reproducible for the same drug. We can do a more stringent test if we have replicates for each set of drug concentrations. So in replica-based cross-validation, we would train on one replicant and test on the other. Of course, there's more than one permutation to try here since we can mix and match which wells belong to which replicates. And that's okay because that will give us some statistics about how well AIs are performing across wells. Both of these, the cross-well and the cross-replicate and just the cell-based cross-validation are, they're both examples of cross-validation, which is an important term to remember. It means that we're testing the AI or using it to score samples with a different set of samples than what we use to train. So we're never actually testing the AI with the same samples that it was trained on. This is the only legitimate way to score samples with an AI, and almost all AI systems are set up to do this automatically. So with replicates, we're getting more into training robust AIs that can be used over and over again and used in production. You would want to use both technical and biological replicates to train them to ensure that you cover all sources of variation. While this kind of training is useful for assays based on target phenotypes or model compounds, it's probably overkill for just scoring compounds based on any observable concentration effects.
Okay. So a quick reminder to those of you participating, please chat questions into the Q&A box for Ilya. So Ilya, since you're not telling the AI to look for a specific thing, what happens if nothing happens? What if there's no response?
Right. So from the confusion matrices for a case like this, in this case, the negative control DMSO and one of the compounds that didn't elicit a reaction, the confusion matrices tell us almost the whole story, because if you compare them to the ones we've seen before, these are highly disorganized. There's no strong diagonal. The samples are called out everywhere on the matrix instead of close to the diagonal. So sorry, can you go back for a sec? I just wanted to point one thing out. So the thing about the accuracies, people sometimes put too much stock in the accuracies here. And the important thing is that you can have an inaccuracy reported in the confusion matrix, even though it's put samples into a neighboring category, right? So it's just as wrong as far as the confusion matrix is concerned to put things in a neighboring category as to put it into a completely different category. So that's why it's important not to get completely hung up on the accuracies. The important thing here to take away really is where the samples are distributed with respect to the diagonal. So let's look at the box plots. Right, so you can see a similar story with the box plot. There's no upward trend in the responses. But I want to point out something interesting, is that, and this is an effect when we are testing on the same drug that we used for training. And the AI is fully confused, so there's no signal, in other words. What you see is that the AI isn't scoring samples near zero. If there's no response, each class looks the same as every other class. The AI's marginal probabilities will then also tend to be the same. When the marginal probabilities are the same, there's no waiting for the average, and all you're just doing is just averaging all of the concentrations. And so the dose response would then be a flat line, not at zero, but at the average concentration. In contrast, if we were scoring samples using an AI trained on a standard curve, the cells from non-responders would look like the bottom end of the standard curve, so they would all score near zero. So this is what you are expect to typically see, in fact, is that when you have no response and you're training on, and you're scoring the same drug as you trained on, the scores will be in the middle of the range and not at the low end. So let's finish off with a summary view for a set of compounds scored by their own AIs so that we can compare their results. So there's no standard curves here, no target phenotypes, no assumptions about what will happen with each drug. You can see the negative controls that were treated just like all the other compounds here. They're green stripes because they're in the middle of the assay range, like I just explained. The ones that have a response are blue on the low end and red on the high end. You can tell a difference in the responses between the different drugs. For example, comparing the positive control mercury compared to rotenone.
Okay, so say you're in a lab and you have thousands of compounds to screen. How does AI-enabled HCA help with that bulk?
So first of all, as we've seen, the big difference with AI compared to conventional image processing is that you do not have to define to the computer what the response is morphologically. You don't even have to know if there will be a response. All you need to do is tell the AI what wells belong to what concentrations, and the AI will tell you if it can differentiate between them. You can use these outputs for quantitative reproducible assays, and you can build in as much generality into the AI as you need to accomplish your goals. Of course, up until recently, this was a bit of a false promise because we gave up on having to define for the AIs what they need to look at, but in exchange, we had to fiddle with training parameters for very complicated AIs. So we didn't really save ourselves that much work. We just switched specialties. So recently, this has also changed, and you can see from the screenshots I showed of ViQi's web-based user interface that there are no real parameters for training the AIs. The best practices for AI training, the models that have historically performed well with this kind of data, and the parameter sets that are typically used are all automatically evaluated for you by the machine. And you don't need to set any parameters on that. This kind of training requires tremendous computing resources. The ability to do this all in the cloud means that you can bring together a great deal of computing for a very brief period of time, very cheaply, in order to do this automated training. Since the user interface is all browser-based, there's no software to install or computers to maintain to do all of this in the cloud either.
Okay, reminder that Ilya is taking a few questions at the end, so please add yours to the Q&A now. So Ilya, do you have parting thoughts for us, things that people should remember and take away from today?
Yeah, so as I mentioned, you don't need to use AIs, you don't need to be preoccupied or have preconceived notions really about the phenotypes that you expect to see in order to use the AIs to develop quantitative assays. And like we've seen, you don't even need to know if there will be an effect in order to use an AI. A confused AI will tell you that it can't see an effect. With when you successfully automated the AI training, then it starts to look very non-technical. Conceptually, the kind of AI experiment setup is very similar to how you would do typical experimental setups, and it has the same considerations. And it's actually much more similar to how we think about biological experiments than when we process images using conventional image processing techniques. So just to reiterate that, what I mean by that, we saw how you would use positive controls or standard curves to train AIs, and you always compare the results against the negative controls. You have to account for sample bias and sample variability when you're training AIs, just like for any proper experiment. And you can't allow your controls to contaminate your unknowns when validating and testing your AI, just like in a regular experiment. So good experimental practice is really all you really need to effectively use AIs in quantitative imaging assays.
Okay. So a couple questions are coming in. Thank you for chatting them in, you all. First question is it possible to use these tools to find similarities between the effects of different drugs?
Yes, so this leads a little bit into a potential topic for an upcoming webinar. But what we've seen with the different concentrations that the AI can tell you about the different concentrations, and it basically senses that some of the concentrations produce similar effects to other concentrations. And so you can actually use that to look at phenotypic relationships between drugs. And so, or actually, for example, RNAi screens. So we can train an AI on each compound independently, just like we're training the AI on each drug concentration independently. Except here, you would use the drug's effective dose to compare the effects. And so now we're talking about comparing the outputs of these independently trained AIs in order to gauge phenotypic distance between the effects caused by these compounds. And this is possible to do, and we can discuss how to do this in the upcoming webinar. This is extremely useful, for example, when you're doing target identification using RNAi libraries or other genomic kind of screens, because you can see, for instance, if you hit the same gene in the same pathway, right, with two different RNAis, that it should produce a very similar phenotype. And even though you trained an AI and said that these are different classes, the AI will confuse them, and it will tell you, no, they're actually very similar. And the similarity tells you that we must be hitting the same pathway and producing a similar phenotype. So that's a topic that there's a lot of exploration, there's a lot of state-of-the-art research in AI, how to go about doing this kind of thing. And these are some of the things we can address in the next webinar if people have interest in that.
Very cool. So there's a question from Oksana in the chat. I'm a bit confused about dose-dependent classification. How do you calculate probability of positive cells from positive-negative training, or do training on each concentration separately? And if so, and do training on each concentration separately? And if so, does it match between experiments?
I think I understand the question. I'll paraphrase it. So I think the question is, how can we, where do we get the marginal probabilities from in order to compute the score? So the marginal probabilities are the direct output of the AI. The AI was trained in the example here on, I believe there were 6 concentrations. So there are 6 classes defined for the AI, right? And 1 concentration per class. And so the AI gives us a probability distribution, 6 probabilities, and we multiply them out. So we know the drug concentration, the AI gives us the marginal probability, we multiply them out, we get a score, right? If we want to classify the sample, then we can just say, you know, that's the one with the highest score is the one that we're going to put in a category, the corresponding class or concentration. I hope that addresses that. If there's good reproducibility between the concentrations in the different wells, for example, when you have replicates, then you should be able to train an AI on one set of wells, right? And then use it to classify a different set of wells. Typically, though, you need to train an AI on many different combinations of these kinds of wells in order for it to be robust enough to overcome the inevitable experimental variations from well to well. Even when we're talking about the same drug with the same concentration, there's always experimental variation. And so you need to train the AI with that variation so that it will be able to generalize to a new Hopefully that makes sense.
Okay, you froze for a second then, but I think we got you. So I have an anonymous question. Do you do any cell segmentation before applying the model or during the training? And if so, what do you do?
Yeah, so I kind of skipped over that. We covered that a little bit in the previous webinar. So there, so ViQi uses proprietary cell segmentation to identify cells. It's quite robust against different cell types that we've tested it on, at least. But it's a combination of fairly conventional techniques. But we also we train AIs to do segmentation. And that's actually another potential topic for an upcoming webinar, is how do you use AIs to do really fancy segmentation that, is not very amenable to traditional techniques. That's also possible, and that's something that we can address. But definitely we're doing segmentation here, and we're making all these calls that I've talked about on individual cells that were segmented. Now, I want to add also that there's a way to do this where you don't actually have to do segmentation. The reason you can get away with this is because in these kinds of images, the cell fields are kind of self-similar, right? So if you just took random tiles from the whole well, basically all the tiles would look the same. And so instead of carving out individual cells, you can say, well, we're just going to look at tiles. So we'll just take random tiles and train the AI with that. And it's actually quite effective. So if you have, for instance, a mess on your hands because your cells all swam together and that's the only way they're happy, and it's hard to segment them, it is possible to do all of this without doing any segmentation. Of course, the calls then would be on individual tiles and not on cells, but when you average them together to get a score for the well, it would essentially mean the same thing.
Okay. So someone asked, how can we determine what the AI is looking at to differentiate the classes?
I have a slide for that. I'm glad you asked. And the reason is because everybody asked that. And so let me adjust the thing here. So one of the outputs that we produce, because everybody asked this, is sort of a scoring of the components that the AI used to make its call. So to get a little bit into the detail of this, so one of the AI technologies that we use is based on image features. What image features are, they're numerical descriptors of image content. They're sensitive to various things like texture or just various statistics. There's literally thousands of them. So part of the AI training is that the AI has to figure out a way to select features that are actually important for differentiating the classes. And so it scores all of them. And so when you can look at the scores that it assigned to the features, and from that get a potential understanding of their relative importance. So we've categorized them into various categories. So first of all, oops, can we go back to that slide?
Sorry.
That's okay. No, is it being stubborn? That's okay. But what you can see is that the results provide you indication of the relative importance of these features based on their scores. We can categorize them by channel, because we, of course, compute these features on every single channel in the image. And the examples we've seen, there are 2 channels, but you can have 5 channels or 500 channels. Why not? And so you can simply look at what channel is the signal in, right? Is it mainly DAPI or mainly the viability stain, right? Here you can see it's kind of evenly split, right? The other thing you can do is the feature algorithms. They're kind of different types. Like I mentioned, some of them are textures, some of them are geometrical, um, others are essentially pixel intensity statistics. So there are several categories you can define, and then you can sort of report with their relative importance as categories. You can also, of course, look at the individual feature scores, the individual algorithms. But there's a really important thing to keep in mind here. And that is the features and these kinds of AIs, they work in concert. So, and they work in large sets. And we're actually not very good at working with large sets. We tend to think of, there's 5 variables and, these 2 are the winners and the rest don't matter. And that's not how this works at all. And so our intuition is really not very good here because the AI is using several dozen of these features that it scored, and each of them contributes very little to AI performance. If you eliminated a few of the most significant features, others will take their place. In fact, most AIs that make use of dozens of features is usually impossible to statistically detect any change in AI performance from eliminating individual features. So there's no such thing as the most important feature. And so you can sometimes gain some insight about what the AI is looking at, but more typically, it's not a very productive thing to look at. Better to make new experiments to probe mechanism than to try to shortcut around it by looking at features.
All right. The last question, the features used for classification, are they crafted specifically by ViQi or are they generated by the AI?
Well, so it depends. So when we use what people call deep learning, convolutional neural networks, there are various degrees of coming up with their own features. Typically, in order to be able to use the small kinds of data sets that you typically find in biological experiments like this, you really need to start with something. So even if you use deep learning, for instance, you would use transfer learning. So some neural net that was trained on, dogs versus cats, to use a silly example, versus cars versus airplanes. And there are features in the first few layers of an AI like this that can be used for microscopy or for astronomy or for whatever, because it essentially does the same thing, which is that it produces numbers that are sensitive to certain kinds of image content. And then subsequent layers of a neural network would then put these things together in various ways in order to produce a result. So this is an example where you're constructing features de novo. But we have 5 decades of digital signal processing literature. There's a lot that's been done, and there's no real reason to throw it away. And so there's a lot of ways that we have, algorithm techniques to reduce pixel content, 2D pixel content, down to numerical values. And this is why we use a bank of thousands of them, because we have an effective way of reducing doing the dimensionality reduction from thousands of features down to a few dozen, depending on the specific problem that you're training the AI to resolve. So it's a combination of a very large bank and an effective dimensionality reduction technique that allows you to use um, essentially the same bank of features to process any image. I mean, in my when we were first characterizing these, we kind of went all over the place to see what kind of images can we process. And so we developed this to look at microscopy images, but of course we can do face recognition, we can even analyze modern art, um, all using the same features. So the feature selection was done at the beginning to come up with this huge bank, and then the automation of the AI training selects out of that standard bank the features that are most useful for a particular problem. So it's a little of both.
Awesome. I can't wait for the Mondrian feature set to drop.
Well, I'm sure, yeah, we looked at abstract expressionists versus impressionists versus surrealists.
Fun.
Okay. And speaking about phenotypic distance, so the impressionists and the surrealists were grouped together by the AI pretty effectively. Probably you would predict, so like looking at Rothko versus Kandinsky versus Pollock. At one end of the spectrum, you have Pollock. At the other end, you have Rothko with Kandinsky in the middle. So the AI does a pretty good job of recapitulating what art history teaches us.
Fun. Come to the webinar for HCA and leave with some modern art knowledge. Love it. Well, thank you so much, Ilya, for all of your insights today. Thank you to the ViQi team for making this happen. And thank you all for joining us. We are here to support your HCA needs, so feel free to reach out and follow up by email. We will email you later today about the opportunity to join Ilya for a half-hour consult, so watch out for that. And please don't forget the survey that will pop up as soon as you close Zoom. So have a great day, stay safe and well, and thank you for joining us.
Thank you very much.